We're going to get right into it today. We want to talk about those formal reports, so we want to talk about the midterm. So let's talk about the formal reports first. So we've been through the title, the introduction, the methods, and the results. So that's the first part. Make sure you do each of these sections, and each section should have a little section title, and they should all be in paragraph form, except for the graphs, of course. They're graphs, they're images. So let's talk a little bit about the results, or sorry, the discussion section. You can see these words I'm using constantly, right? I'm talking about being specific, thorough, complete. So this is the part that, you know, to put it bluntly, you want to be verbally, you know, very verbose and long-winded, and you want to tell me about everything. So kind of the main thing you're doing with the discussion is there are questions at the end of Lab 3, and the questions ask you, for example, to compare the results to your hypotheses. They ask you to, you know, explain your results. You're going to look at your, for example, your temperature experiment, and you're going to talk about, okay, well, we see that at the highest temperature, 85 degrees, we have a lot of membrane damage. At the minus 20 degrees, there's a lot of membrane damage. So you want to talk about that stuff. You want to, you know, really just look at it in detail, and why, like, why was there so much damage at you know, 85 degrees, right? So those are all the things that you need to discuss. And you can see that note I have there is that your job in the discussion is to convince me that you get it, right? Convince me that you understand what is going on. So paragraphs, organized, thorough. You know, your discussion, I'm thinking for most people, it'll be minimum of 2 pages, probably closer to 3. Again, it depends on how verbose you're going to be. But I'm not going to dock you if you have too much. So make sure that you are thorough. I have some sample sentences here for you, just some reminders to be very, very specific, right? You can see the first sentence. Clearly, temperatures below freezing are damaging to beet membranes. Okay, yeah, that's not too bad. But let's expand that, right? Let's turn it into a whole paragraph, and let's give a lot of details. So below freezing, well, okay, what are the details there? Minus 20. Okay, that's a very specific detail. How damaging, right? You know, look at this value, 16 micromoles. Now, that's not necessarily the value that you're going to get. I just threw down a number. But compare that number to, you know, let's say the room temperature treatment. And we can see that it's, you know, we could talk about it's being 10 times as more damaging, or, you know, something like that. Give me some context, right? Just saying something is damaging, well, I don't know. I mean, maybe I don't know anything about membranes. You can see the last thing I've done here in this sentence is I've referred to the actual data. That's important too, so that, you know, you can discuss it. And if the reader is like, okay, yeah, that's interesting, where can I find more? Figure 2. So make sure you're specific. And like I said, you know, don't feel free to be long-winded, but do make sure that your information is accurate and organized and clear. This is the kind of thing where if you have a friend or a mom or brother or sister who can proofread it for you, then it's not necessarily a bad idea. They don't necessarily need to understand what's going on, but you know, if they can proofread and make sure that your sentences at least, you know, seem coherent and are grammatically correct, those kind of things are going to be very, very helpful. You can see I'm continuing. I'm not just leaving it one sentence. I'm explaining why. So, you know, why are temperatures below freezing damaging? And this is where you may have to do a little research in your textbook, or maybe, you know, a little bit of time on the internet may help you to find out what is going on. You can see the last thing I have, and maybe I'll just make a quick note about referencing. I was going to talk about referencing again next Wednesday. So you are going to have to cite 2 things in your lab reports. One is you're going to be citing the lab manual, okay? And the lab manual must be cited in the methods section. 2, you need to find a journal article, which you did for lab 2, so you should still have a journal article. And doesn't matter to me how closely related it is to beet membranes. I'm not going to read the journal paper. but what I want to do is I want you to see you cite it and reference it properly. Now, it can be cited and referenced in the introduction or cited and referenced in the discussion. It doesn't matter, as long as it's cited somewhere. If you cite it multiple times, that's even better, right? You know, the whole thing behind referencing is, you know, twofold. One is you don't want to be taking credit for something that is not your stuff, right? That would be plagiarism if you were doing that. Secondly, it adds authority to your report, right? You just telling me that, you know, freezing is damaging because, that's great. But if you have a study to confirm that, then it sounds a lot more authoritative. And this is very, very important, and becomes more important as you, you know, get into your second year and third year and write more reports. In this report, like I said, I'm just looking for 2 citations. You can do more, and that's great, but there better be a minimum of 2 citations for this report. So I just wanted to make a point about the chemistry of what is going on in the solvent part here. Okay, sometimes I get students talking to me about acids, and I'm just like, where is that coming from? Because I do not see a single acid in the solvents. These are not acids. Water, methanol, ethanol, propanol, these are not acids. These are alcohols, right? Alcohols are not acidic, not in a biological context anyway. And so what is going on here? This has to do with polarity. So you can see at the bottom, I've thrown down a fatty acid for some context. And notice my fatty acid. Hopefully you know that fatty acids are very, very nonpolar. And that's because they have all of these carbons and hydrogens. And so that's actually what's going on with our solvents. We're adding more carbons and hydrogens, more carbons and hydrogens. And the carbon-hydrogen bond is it's a non-polar covalent bond. So the more non-polar covalent bonds that you add to a molecule, the more non-polar the compound becomes. Or you could also say hydrophobic, of course, right? So that word is used interchangeably, hydrophobic. That's an R. I have a hard time writing R's with this pen here for some reason, this computer pen. Okay, so one more quick comment. Human error, right? You can see here is a human error with some sort of railroad construction that looks awful. But so I know one of the questions asks you about, you know, what can be improved and all that. And I just want you to avoid discussing human error, okay? That's like, you know, grade 6, right? Of course we make mistakes. Every time I pipette something, you know, there's going to be a little bit of error, right? Every time you pipette something, there'll be a little bit of error. And if you're really inexperienced in the lab, there's going to be more error because maybe you're not as familiar with the pipette. Or maybe the, you know, there's all sorts of hypothetical things going on in terms of maybe my thermometer isn't measuring properly, or, you know, the water bath, when I used it, it could have easily been, in fact, I think I showed you in the video, it was 64 instead of 65 degrees, right? So there's always lots of little error in there, all right? I think, you know, the question is asking you what you can do to, you know, improve the experiment. So, you know, think about how the experiment was done and what we can do to, you know, maybe test the hypothesis better. You know, if you've taken statistics, you know, what you can do to kind of beef things up. And, you know, I'm trying to talk around this without necessarily giving you the full answers on this. But we can make this experiment better, right? You know, your hypothesis around, you know, temperatures, think about, you know, the temperatures we use. Could we do better than that? Could we use other temperatures? So I'll let you think about that, but don't go on about human error, because I understand that that is an issue, right? Let's go beyond that, please. Okay, so we're going to do the midterm review. I'm still, you know, putting the final touches on the midterm here, but I think the final midterm is going to be a little bit more like this. So one long answer question, and I've actually expanded the matching part of the midterm. So I think that's going to be the final thing. It will be worth 35 marks. And as I mentioned, it's on Moodle, and I'll show you, you know, some sample questions here today, and I'll show you a little bit about the Moodle interface for the written questions. So just a reminder about the lockdown browser. I think last time I checked, there were about 3 students that had not tried this yet. Okay, what I do not want to have happen is you know, our midterm is on, what is it, 4 o'clock on Monday? I do not want a phone call at 3:59 on Monday where somebody is panicking because they can't get their technology to work. This is your responsibility. If you can't get your technology to work, or if your webcam is broken, you need to buy a new one or borrow somebody's computer or do whatever it takes. Okay, now I think, like I said, most of you have done this and tried it out, and you see what's involved with the lockdown browser. You know, check in 5 minutes before the test so you can actually set this up and you're ready to go, and you can have the full hour to do the exam, right? So if you're not setting things up till 4 o'clock and then, you know, you're having to start a few minutes late, you're not going to get the full hour to do the exam. So it's really important that you do this ahead of time. I do not have it programmed to ask you for your ID. I think we're being told we have to do that for the final exam, so that will just be one extra step we'll do. Maybe we'll try it for midterm 2 or something like that. If you are having some technical difficulties, let me know. I might have some suggestions. We do have somebody at the college who's really familiar with the lockdown browser technology, and she can offer some suggestions. If you're having issues with your Wi-Fi or finding a quiet place, I might be able to find you a place on campus where you can access better Wi-Fi or something quiet. But if there are those issues, let me know. Like I said, I do not want any phone calls 3:59 on Monday. So believe it or not, I had a midterm yesterday at 10:30 a.m. and somebody phoned me at 10:29 a.m. What am I supposed to tell the guy? You know, it's like I can't help you if you're going to call me a minute before. Okay, so hopefully this does work well for everybody. Do make sure that you're, you know, you do not have clutter around, because it was an environment check. I want to see that you're not having all your, you know, your books and your notes nearby. And do make sure that you're well lit. I know another instructor had an issue where somebody was writing in the dark and all they saw was a black face, and they couldn't even tell if it was the right student or not. So you got to make sure it's lit well enough for that. Yeah, study hard. I mean, it could be a lot worse. You could be studying for something else, but instead you're studying for cell biology, and that's actually a big bonus because this is your favorite class, right? I hope, anyway. Okay, so I'm going to go through these topics. You can download these. I've taken these PowerPoints and I've actually added a few other things to them. I haven't uploaded the updated ones, but you know, if you're interested, send me an email. I can forward you the new file. I'm just going to assume you can read. I want to get to some of these sample questions right away, but topic 1, you can see we're talking about the scientific method and a few other things like history. So here's a sample multiple choice question. So you can see that, you know, just some tips around answering multiple choice questions. Go through every solution, make sure you understand it. And unfortunately, we don't have the opportunity, you know, to be writing with a pen, because what I like to do when I'm writing multiple choice questions, you know, I like to go through and read everything and cross out the ones that are incorrect, right? So, you know, for example, the independent variable is not the control. No, control is not a variable, right? But one thing that you can do in the Moodle system is you can flag questions. So if there's a question you want to go back to and reread, there's a way to actually mark it and flag it and go back to that question. So let's take a look at these. Is the factor that is deliberately manipulated? No, that would be the variable, the independent variable. Yes, that is actually the correct answer. I shouldn't have put it at A. So the independent variable, sometimes we call it the manipulated variable. And we also have the dependent variable, which is sometimes called the responding variable. So just something to think about. Dependent means it's dependent on what you did for the experiment. The dependent variable is the thing that we're measuring. So the independent variable is the thing that you're actually manipulating so that you can get results. So that's actually the correct answer, but let's just go through the other ones just in case. Something which a change is observed. No, that's the dependent variable. It's not altered. No, variable means you're changing something. Has no impact on the results. Of course it doesn't. Oh, that one's wrong. And there's the correct answer. There's going to be some true and false in there. I think I have 4 or 5 true and false. They're going to be mixed in with a multiple choice. So here's a sample true and false. He was the first person to observe cells using a microscope. And The answer is no. Actually, Robert Hooke, remember Robert Hooke was the guy who was looking at the cork cells. He observed the cells. Then Leeuwenhoek was the first person who observed bacteria. So do make sure you read the questions carefully. Question 2, macromolecules. So we have our lipids, our carbohydrates, our proteins, and our nucleic acids. And so I have some sample questions here. It says, you know, what are the following structures? I just want to spend a little bit of time going over these structures and kind of reviewing, you know, some principles about them. And so you can see these ones here. You probably know the one in the upper right already because we were just looking at that a minute ago. So let's label that one a fatty acid. So how do I know that's a lipid or fatty acid? So we have all of these carbons and hydrogens. So that's the first part that makes me know that it's a lipid. A fatty acid also has this group here, which I know is a carboxylic acid group. So I know some people may be a little rusty with their chemistry, so I'm trying to go over these a little bit and you know, give some practice for you. Okay, so let's just start. Let's take a look at the one. Let's take a look at the one below this. So you're probably wondering what that is because it has, let's see here, we've got 1, 2, 3 carbons on it, but then, hey, we have these alcohols. So it looks like the carbon and the oxygen are 1 to 1. So that's kind of an indicator that this is a carbohydrate. And actually, since it has lots of alcohols, that also is an indicator that it's a carbohydrate. So this here is definitely a carbohydrate or a sugar. I guess sugar is the option up top. This molecule, in fact, is glycerol. So you may remember glycerol as being part of the triacylglycerols, right? And glycerol is in the name, but it is technically a sugar because it has, like I said, a 1 to 1 carbon and oxygen ratio and has those all those alcohols in it. So if you take a look, I'm looking around here and I'm seeing, okay, what do we have here? That's not an alcohol, that's a carboxylic acid. So I'll come back to that in a moment. But this compound here, look at that. Look at all those alcohols all over the place. In fact, same thing over there. That particular molecule has a 1 to 1 ratio of carbons and oxygens and lots of alcohols. So this is a sugar. In fact, that is glucose. Anything else? Any alcohols? I see 1 alcohol over here, but just 1. That's not enough to really, you know, do much to that particular compound. So let's talk about that one I have circled that has an acid group on it. So this is an acid. And this here is an amine group. So hopefully just right from those words, we have an amine and we have an acid. So that particular compound is an amino acid. Okay, working through these. We have a couple left. So this one has its little alcohol. I told you 1 alcohol and a big giant molecule isn't so much. But the big indicator here that we're seeing are these 4 rings, right? We've got 1, 2, 3, 4. And whenever I see anything like that, it's a sterol or a steroid. So that's actually a cholesterol, that particular compound. Okay, one more. You know, maybe we could match the remaining word to the remaining molecule, and it's a nitrogenous base, and the indicator here is we've got all these little nitrogens, right? So this is a nitrogenous base. I see there's a question there. I'll get to that in a second. Nitrogenous base. There we go. So someone says, if you call the amino acid compound a protein, is it considered incorrect? That's kind of a gray zone. Usually we say a protein is a string of amino acids, right? But you know, if I were to classify this as any type of macromolecule, it would fall into the protein category. So you know, it's kind of, like I said, really depends on the question. But usually I'll try to be a bit more clear because it is specifically an amino acid is a component of a protein, right? Because a protein is a polymer of amino acids. Okay, so I have a couple more for you. These were all, I was looking for smaller structures. In the next slide, I wanted to show you some larger structures. I'll just keep the chat function open if anyone has any other questions. Okay, what do we have here? Any alcohols? That's the thing I'm looking for, right? So down at the bottom, look at, we've got all these alcohols. Oops, that always does that to me, this little pen. all these alcohols here, lots and lots of them. So that's got to be a carbohydrate of some sort for sure, 100%. Do I even have that as one of the options? I do have that. Of course, I can't spell carbohydrate in my slide. Okay, well, I'll fix that someday. Okay, the other thing about carbohydrates, by the way, and I think I mentioned this before, is that often you're looking at, they like to form these hexagon and pentagon structures. So does it have hexagons or pentagons and lots of alcohols? Definitely a carbohydrate. Okay, any other alcohol groups? Okay, the one in the top left has a couple, right? It's got an alcohol there, another one there. Not a ton of them, so probably not a carbohydrate. Although this little group here in the middle, I'm just going to put a D for deoxyribose, because that's deoxyribose, and so it is a carbohydrate, that little structure there, but it's part of something bigger. And that little carbohydrate, that pentose, that 5 sugar carbon, has been attached to a few other things. So we have a nitrogenous base here, lots of nitrogens. We have some phosphates, and those are the 3 ingredients of nucleic acid. We have phosphates, a pentose sugar, and a nitrogenous base. So this is a nucleic acid. Okay, speaking of nucleic acids, hopefully everyone recognizes the one on the right. There's an easy one. We have DNA, that's the DNA double helix. So of course, this is a nucleic acid. There we go. Okay, so what else do we have left here? We have this thing in the middle, and we have this thing at the bottom. So this thing at the bottom here, let's take a look at this. This cartoony structure. This is a protein. I know you don't see any atoms on there, but the proteins have so many atoms that we're trying to represent it in some way that's a little easier on the eyes to understand what's going on. So you can see those little swirly things, those are alpha helices, and they're trying to color code it. I don't know why they have green and blue, but I think that actually looks like 2 proteins that are merged together, so it would be a dimer. So that is a protein. So I showed you a few pictures of proteins in our lectures. Make sure you know what they look like. Obviously, they're going to be different from DNA or RNA. DNA is usually in that nice little helix, which is a good indicator of what you have there. Okay, the one in the middle. I'm looking at this and I see, you know, all this N is fun. I see all of these carbons and hydrogens, so we definitely have some sort of lipid thing going on right there. I have this group here. This is glycerol. I know it looks different from the other glycerol. That's because it's attached to some various things. We've got, looks like we've got some ester bonds in there. And so in my head, I'm going back to that table that we made, and I'm thinking about ester bonds. And if you remember, we were looking at linkages, and ester bonds are found in lipids, and particularly triacylglycerols. But if I look at this, I have 1, 2. This is not a triacylglycerol. And the last part of this is I do have a polar group up there. And that polar group, you can see, has a phosphate in it. So this one here is I'm not going to write the whole word phospholipid. I didn't really leave myself a lot of space, but I'll make a little arrow for you there. That is a phospholipid. So hopefully that was helpful. Like I said, I want you to be able to look at these molecules and identify what we have. So like I said, they all have some different characteristics, and again, you'll have to review the notes for some of the examples. Okay, so let's move on. Topic 2, we were looking at mostly microscopy and a few other things. And so you have to review those things. And so what I want to do now is show you a sample type of long answer question and talk to you a little bit about answering these types of questions. So you can see this says compare and contrast light microscopy with the electron microscope, and give some examples of the things you are going to observe. Okay, so this is going to be a 5-mark kind of question, and I know I have it. Where was it? So here's a sample of what that might look like in the Moodle midterm format. So any of the written questions are going to look something like this, right? So it'll have the question at the top, and then you have this big blank window to answer things in, right? So this is a 5-mark question, right? So you want to give me enough to give you 5 marks, right? You know, I've seen this a lot. I get the 3-mark answer. People have like 3 points. If you want the full 5 marks, give me lots. The other thing that's very helpful is if you organize your answers. Okay, you can write things in paragraph form. That's fine. You know, maybe, but what's really going to help me to grade it is if you you know, break it into sections, right? You know, so you could write up here, and you could have you know, maybe light microscopy. Let's say we could put up here. Okay, and then, you know, we could even use some formatting if we wanted to. You can see up there, there's a, you know, I could bold it or something if I really wanted to, and then I could start writing. And maybe I could, you know, again, to make this easy and organized, I could have points, right? So the first point is, let's say in this case here, the illumination source. The illumination source is light, and I could give some examples. I could say UV or visible light, right? So that would be enough for half a mark, right? Because I have to give the comparison part for the electron microscope, right? So the electron microscope, I would type that all out, and I would say that, you know, except for the illumination source, in that case is an electron beam. Another thing that you can do, and I'm going to show you how to do it now, is if you take a look at this, there is a way to make a table. So if I sort of click on this little thing, notice it gives me more options. So now I can add superscript and subscript, I can justify it and all those kind of things. And so in this case here, you can do your answer in a table, and that's a great way to do it as well. So you can see I clicked on that little table icon. I'll show you that again. It's right here. So what I did is I clicked on this, click on the table icon, and then it's asking me how many rows and how many columns. You know, I'm not exactly sure. You know, sometimes when I'm answering these kind of questions, I'm thinking, okay, how many rows do I need? So I said at least 5 points. I'm just going to throw down 8. A few extra rows aren't going to hurt even if I don't fill them all in. 3 columns, so I'm going to have kind of the feature that I'm talking about, and then light microscopy and electron microscopy. Or you can do 4 columns if you want to do light, SEM, and TEM, right? So it's up to you how you decide to do this, but I'll create the table. Maybe you can see it there. Sorry, I see somebody has a question. So someone says this type of question does not need to be in paragraph form. No. Point form is actually much easier for me to grade. And like I said, you can do a table. There's different ways to answer these kind of questions. Like I said, in this case here, I'm just showing you how to do it in a table form. So there's my table. It sees here, so I could put maybe light microscopy here, and maybe electron microscopy here. And by the way, on midterms, if you spell things incorrectly, it's not really a big deal if I can figure out what it is. If I can't figure out what it is, if you really butchered the word badly, I can't help you there. But you can see in this case here, I've done a typo, and I can clearly tell that says light microscopy. It's not really a big deal. Formal reports, on the other hand, hopefully you can spell properly, because you do have the chance to spell check and all those kind of things. That being said, right, if your grammar is just atrocious and I have to reread the question multiple times, then I will not give you a perfect grade for that. Okay, so you can see my first idea was illumination source. And I notice the default font is extremely small. If you want to do better, you can always highlight these, and notice up here I can maybe try a larger font size. That's a little better. If you really want to color code it, I wouldn't waste your time color coding things. You do want to do well and get to all the questions. Illumination source, so I can say visible or UV light, electron microscope, electron beam. So that's not too bad. I would give you 1 mark for that, and I think I had said before, some of these questions, this one is kind of open-ended, and there's a lot of things you could discuss. You could discuss how things in electron microscope are stained with heavy metals, and you could talk about in a light microscope things could be dead or alive. Like I said, there's a lot of things you could discuss. Some parts of the question will be required. So when you're comparing light microscopy and electron microscopy, you better talk about light and electrons. I want to know what's the deal there. Okay, that's something that's essential. Notice in the actual question as well, it says something about providing examples. So do provide examples. Do not give me vague examples. Don't just say example, a cell. Yeah, that's great. An egg is a cell, right? An egg that you eat for breakfast, that's a cell. I don't need a microscope to see that. but you know, what kind of cell? What specifically are you looking at, right? So light microscopy, for example, a good example would be a gram-stained bacterial cell, right? You know, you're seeing something, you have an idea how big a bacterial cell is and why, you know, what exactly you're looking at. In a gram stain, we want to see, is it pink or is it purple, right? You know, the two types of electron microscopy, so you need an example for both. I'm not going to give you all the answers for that. You can look these things up, but I'm just trying to give you an idea how to answer this question. So like I said, 5 marks, there better be at least 5 complete points. If you don't feel like there's enough information there, then add a couple more points. Usually I tell students, you know, when this is written, you know, with a pen and paper, you know, this kind of question is probably at least half a page to 3/4 of a page for the average student. It really depends on your handwriting size and all that. It's a little harder to judge electronically, but do make sure that you do give me enough for 5 points. Okay, so let's go back to the PowerPoint. I have some other questions we're going to try here and take a look at for review. So topic 4, different types of membrane structures. So topic 4 was really kind of 3 parts. We talked about membrane structure, we talked about membrane transport, and we talked about cell walls and the extracellular matrix. So make sure you do cover each of those in your studying. Here's a sample multiple choice question. It says, which of the following molecules cannot easily diffuse across the plasma membrane? So watch out for words like that, right? We want to make sure if we're reading the word cannot or true or false or something like that. I guess it just popped up the answer for you. Charged molecules. Everything else is good. Anything that can fit through that hydrophobic layer, so hydrophobic molecules are good. Tiny things like water is good. Uncharged usually means nonpolar. Maybe not the best choice of word there, but nonpolar and hydrophobic are used interchangeably. So here's one I said we'd come back to for the review. So I'll go through this quickly. I'm not going to be too thorough on this because I do want to make sure I get through everything for the day. But thinking about what these 3 things are, right? So the cell membrane, for example, what is this made of? This is made of phospholipids. Phospholipids, proteins, sorry about that, I wrote too large, and carbohydrates, right? What type of cells have this structure? All cells have membranes. And the functions, I really don't have a lot of room there, but we'll put a little bit of room there. Enclose the cell, and the other big one is transport molecules in and out. In and out. Usually I do this on a whiteboard, and I guess I wasn't prepared to, didn't leave quite enough space there. But anyway, like I said, we're just going to do this one quickly. Cell wall. Okay, what's the composition of the cell wall? Well, it depends on who you are. If you are a plant, then it's made out of cellulose. If you are a bacteria, we're made out of peptidoglycan. If you are a fungi, you're made out of chitin. So, you know, lots you could write there. What types of cells have these structures? Well, like I said, you know, the big ones to remember are plants, bacteria, and fungi. And all of those we looked at in some detail. Functions, so cell shape and protection. And protection against what? The big one is osmotic pressure. Remember, plant cells, they don't burst when they're in water. So osmotic protection is good. Okay, extracellular matrix composition. That's a sticky one because it kind of just depends on what cell you're looking at. You know, some cells we're looking at cellulose, or not cellulose, sorry. I meant to say collagen. Sometimes it's minerals if you're talking about bones. So a variety of things. And this is the kind of question that's a little bit open-ended, a little hard to ask you in an exam because I get a whole bunch of different things, but I just want to review some of these things. What types of cells? Some animal cells. Sometimes people get a little confused about the difference between a cell wall and a cell membrane. Remember, a wall is a hard structure and a membrane is something that's fluid. It's got that fluid mosaic model thing going on. Functions, I'm just going to say connective tissue. but sometimes protection if you're talking about bones. Okay, a couple other questions here. We want to talk about some cell structures, and this is going to be a big part that I encourage you to spend a little bit of time reviewing these things. I know people get them confused, and I'm not going to ask you to label something, right, like this. This is labeling something that's more like high school. I want you to know what these things are and what they do. So let's review these things, what they are and what they do. So number one are these finger-like things. Those are made out of proteins, and those are fimbriae. And remember, the other word is pili. Pili is many, pilus is one. So those are attachment. Those are used for attachment. Remember, we also had the sex pilus, which is involved in conjugation. So number two, it's a little hard to tell what number two is pointing to because it looks like cytoplasm, but you can see cytoplasm is already labeled. So number two is actually a ribosome. All right, ribosomes are for protein synthesis. So number three, we've got this lump of DNA. It has a name, and the terminology used for that is the nucleoid. Number four is a plasmid. What is a plasmid? A plasmid is kind of like a mini chromosome. It's circular like the big chromosome, but it's just a lot smaller, only a few genes, like usually 5 or less genes on there. Speaking of 5, what is number 5? Number 5 is a flagella. Actually, flagellum is one, flagella is many. And that is involved in motility, which means swimming. So let's take a look at 6, 7, and 8. So starting at the inside of the cell with number 6, and we're moving outwards. So number 6 is what all cells have, which is a plasma membrane. You can use the term cell membrane, but sometimes that gets confused with other membranes. So we use the word plasma membrane usually because it encloses the cytoplasm. So what's outside the membrane? Outside the membrane is the cell wall, and in bacteria, we're talking about peptidoglycan in particular. So if this is gram-negative, it would have a second membrane called the outer membrane, but this looks like it is not a gram-negative, it's a gram-positive. And so that stuff on the outside is a capsule. Remember, there are other names for capsules. Sometimes they're called slime layers if they're more loose. Slime layers. And the term for both of them is a glycocalyx. And that means sugar coat. So lots of structures there. I have a review of some eukaryotic structures as well here in a moment. There they are. I'm not going to get, like I said, not going to have you label anything for me, but I want you to know what these things are. So let's take a look at these. I have some functions, and so I've tried to not think about these functions quite as obvious, right? I want to think about them in terms of detail. So you can see the first one, it says translation of membrane proteins. So now you got to remember what translation is, right? So that's why it's important for you to know these terminology. Translation means protein synthesis. Okay, so that's easy. It should be ribosomes, but then take a look. It says membrane proteins, right? So in this case here, I do mean ribosomes. Half a mark. And these are ribosomes in the rough ER. So sorry about the lack of space again. So remember, the rough ER has ribosomes, and those proteins are becoming membrane proteins, or they're going to one of the endomembrane systems. So anything that's becoming a cytoplasmic protein, those ribosomes are in the cytoplasm, and the other ones are in the rough ER. So there's just some details there. I want you to make sure you're reading things carefully and giving me lots of details, making sure you're reading every single word. Because I would just say translation of proteins if I just meant ribosomes in general. Cytoplasmic proteins. Okay, well, okay, I guess in this case here, I've already told you the answer. So cytoplasmic ribosomes, or sometimes they're called free ribosomes, meaning they're not bound. So there's another term for you. Number 3, ribosome subunit assembly. So where are these things put together? Remember, ribosomes are made out of ribosomal RNA. Sorry about my little R's. I just have a hard time. I guess I'm sloppy the way I make them. Let me try that again. A little R, a little stick, and a little R. ribosomal RNA plus proteins. So these are assembled in the nucleolus. What is the nucleolus again? It's the dark thing found inside the nucleus. And actually ribosomal RNA is being synthesized there like crazy, and that's why it stains so dark. And the proteins have to get imported, they go through nuclear pores, and they come together and form the ribosomal large and small subunits. Okay, number 4, supports the nuclear membrane. Okay, we got to remember, what am I talking about here? These are some fibers, and they are called nuclear lamina. Number 5, where are proteins glycosylated? So it turns out there's 2 answers for this. They are glycosylated. Glycosylated, oops, that meant to be an R. They're glycosylated in the rough ER. So glycosylate means adds a carbohydrate, and also the Golgi. Okay, number 6, breaks down old organelles. So think a little bit, we did talk about a process, and the process is called autophagy. And that's the process where old organelles are getting digested. So what's the name of those digestive compartments? They are the lysosomes. Okay, one more. Just enough room for one more. Had to give you an easy one. Photosynthesis. Hopefully everyone is thinking chloroplast. So there we go. So a couple more things to talk about for the final exam here. Like I said, for the written questions, this is the part that I find people struggle with the most, is that you need to be thorough. And obviously you need to know your material. And that's where I think sometimes people struggle. You know, multiple choice, it's easy enough to just answer B. But if you don't really know what you're talking about, and that's why I have the written components, because, you know, I want people to have that chance to show me that they know what they are talking about, and you know, to really reward the students that are studying really well. So obviously the more material you learn is good, and I think I had mentioned before, do focus on the study questions and the vocabulary at the end. That is going to help you tremendously to get through this part. I think, let me actually have a couple more slides and I have a couple other things to talk about here. So these are the major concepts. Like I said, if you're looking for a place to start, you do want to take a look at these major concepts. And, you know, some of you may find this first midterm easy. Some of you will find it hard. And I think a big part of it, like I said, is just understanding the vocabulary and really having an intimate understanding of things. It's very easy to look at this stuff and think, I saw this before somewhere in high school, okay? But, you know, that's not necessarily going to be good enough. It's really important for you to know it really well. And that will come out when you write the exam, how well you understand the material. And like I said, the vocabulary is a huge part of it. I know there's a lot of nitty-gritty details in there. Do try to focus on the things that you know, the general rule of thumb is the more amount of time I've spent on something, right, the more important it is, right? For example, I mentioned, you know, about ribosomes. Ribosomes are a target of the antibiotic tetracycline, right? I'm not going to ask you about that. I mentioned it once in passing, and maybe you've written it down and maybe you've learned that. That's great, okay? I think antibiotics are really important, and if you go on into a medical area, it's important to know what tetracycline does, right? But then on the other hand, we did talk about penicillin. I don't know how many slides about penicillin, maybe 5, okay? So know what penicillin does. What does it do, right? That's something that's a bit more important, okay? So hopefully as you are, you know, listening to my lectures, you are taking good notes, and you are seeing, you know, what type of things I'm emphasizing on. If I am writing something down in a Word document or otherwise, right, those things are important. I'm not taking the time to write things down because I like typing. I don't like typing, okay? You know, I'm writing it down because I'm trying to emphasize to you that I think this is important. If you do have any questions or clarification with that, you know, always contact me and find out. But like I said, I want you to go home and study. Like I said, it could be a lot worse, could be another class. You know, in this case, you're studying for cell biology, and this is really cool stuff. And after the midterm, we're going to get into some other things, right? Processes like photosynthesis and respiration. And, it's going to get technically more difficult. So some people find midterm 1 harder because maybe this is your first university midterm ever. And like I said, I want you to study, and checking out that sample midterm might help you out as well. Some people find the second midterm more difficult because it's just technically more complicated, right? We're talking about respiration and photosynthesis. Some people find the second midterm easier because they've been through a few midterms and they kind of had an understanding of how much to study and how hard to work. So my last word of advice is talk to your friends, right? And your classmates, and find out, how long did you study for that biology midterm, right? How long did you study for chemistry? And you're going to find different answers. And then ask them how they did, right? If they tell you they spent about 2 hours studying and they had a 34%, and then your other friend, says, I studied all weekend for it and I did really well, you might want to find somewhere in between. Again, it depends on, how well you can commit things to memory. The unfortunate thing about biology is it involves a lot of memory work, right? And for me, actually, believe it or not, that's actually one of my weaknesses. I'm very good conceptually in mathematics and things like that, but memorizing things is just so painful for me. But when you love something, when you're passionate about it, when you want to talk about it, it makes it so much easier. So hopefully, like I said, learning those words is going to help you to want to talk about biology and make the whole process a little bit smoother. So I think I showed you my good luck balloon last day. This is all I have for you this afternoon. So I'm hoping everyone has a lovely weekend. If you have any questions, I will check my email over the weekend and hopefully get back to you and answer any particular questions. And I will be available on Monday. You can try phoning my office or emailing me as well if you have any issues. Like I said, if you have not done that lockdown thing, do it, because I cannot be responsible for your technology breaking down. You are responsible for your own technology. So have a great weekend. Best of luck studying, and I hope that everyone does really well on the midterm. So take it easy. For some reason I can't stop the Zoom meeting. It's not letting me do this. Let me try something else.